Okay, we are good at that. Okay, I think we can get started. I believe students would be joining in slowly. Welcome everyone to our seminar series in the Department of Food Science and Nutrition at Illinois Tech. I would like to welcome Dr. Sims. Thank you, Dr. Sims, for joining us today, and it is my pleasure to introduce him. I have known Dr. Sims when I was student at UF. I took his classes, and he was my one of my committee members on my PhD dissertation. So I have worked with him on some projects as well, collaborated. His lab's main research is on sensory analysis of various food products. We like to go to his lab when we were students because you get to taste food and you get to compare the food. And I am pretty excited for today's seminar, and I believe all the students are excited as well. And I would I would like Dr. Sims to go from here and take the seminar from here. Thank you, Dr. Sims.
Thank you, Aman. That's a very nice introduction. Good afternoon, everyone. It's a pleasure to be here with you. I was just saying I wish I could be in Chicago. I heard the weather's not very nice, but I've always enjoyed going to Chicago, for instance, to the IFT meetings, and hopefully we can start those up again maybe next summer. So maybe I'll get to see some of you in Chicago next summer at the IFT meeting. But what I'm going to talk about today is something that I do every day pretty much, is sensory analysis. So I'm going to give you an overview of how we detect odors and taste briefly, and then talk about the common sensory testing, and then finish up the end with some examples of some research that I'm doing that are involved sensory testing. So that's kind of the game plan. I would encourage you to ask questions anytime if you want to stop me, or I don't know if there's a chat feature in this software or not, but please feel free to interrupt me, and hopefully we'll have time at the end for any questions that you have. So I'd like to make this as interactive as possible, so please feel free to ask questions. I'm not very familiar with this, but I'm going to share my screen, and I hope this will work. Present now. I'm going to do a window, and there's my PowerPoint. I'm going to share this. And let me do this. I think this will work. And then the slideshow. Okay, can everybody see that okay?
Yes.
Okay, good. So I can't see anybody. I don't know whether you can see me or not. All I'm seeing is my PowerPoint, so I'm going to be talking to my slides, I guess. But this is what we're going to talk about, an overview of sensory analysis of foods. By the way, I've been at the Food Science and Human Nutrition Department at the University of Florida for 35 years. I started here in 1986, and things have sure changed a lot since during my time here. So, you know, if you think about it, we all say this so often, this food tastes great. I hate this. We've learned this so many times. Usually every day we talk about something that tastes good or doesn't taste good, or we like this, we don't like that. And we just kind of take it for granted. So there's a lot that goes on in your mouth and in your brain between when you take a food in and when you can blurt out whether you think this is good or bad, or you like it or you don't like it. So that's what we're going to talk about today. What goes on to lead to these reactions, and then how do we measure those reactions? And so the first thing you notice when you're eating, or whether you're tasting a food in a sensory lab or whether you're just eating, you're going to notice the odor. In wine, it's called a nose, but whatever. It's the odor we get before food goes in the mouth, and that's called orthonasal olfaction. And that's very important. It's certainly a very important cue. If there's something wrong with the food, we would smell it. In the case of wine or beer or certain beverages, we're going to smell it and really get a lot of enjoyment out of the odor. And so we're going to notice the odor, and that's a very important part of the whole process. Then we put the food in the mouth, and then we get we call the flavor. That's the retronasal olfaction. Does it taste like a blueberry, a peach, or what is the flavor system? And that's extremely important. And then the taste kick in. So now we detect sweet, sour, bitter, salty. And that's a very important contribution, certainly. And then there may be other characteristics. There may be umami. Some people could classify that as a taste, but it's certainly a sensation. It may be carbonated. It may be spicy or hot. So there's a lot of other things that can be happening. And the brain is processing all of this at once. So each one of these have a different nerve system coming to the brain. And the brain's got to process that. And so number one, has the brain ever seen that food before? So the flavor of a food, is this something totally new, or is this something that you've had before? And do you like it or dislike it? So learning to identify foods is one thing, and then learning to like them and dislike a food is a totally different thing. But the brain has to process all of this and just say, I really like this food or I don't like this food. So it's a lot of complexity to this. And I'm going to go through all of this in a little more detail. So for odor, orthonasal olfaction, the molecules from the food or the beverage are coming off the food and going into the nose. And I'll show you a picture of that in a minute. So they're entering in from the outside world. And we have olfactory receptor cells that are high up into the nose. And the olfactory receptors within these cells are large proteins embedded in the membrane. And I'll show you a picture of that in a minute. And the receptors bind to a specific part of the flavor volatile. There's not a specific receptor for a specific molecule per se. So what it is, these receptors, these protein receptors bind to a certain chemical structure on the flavor volatile. And then once that nerve, that establishes a nerve impulse, once that has occurred, and then the brain is interpreting the pattern of all of these receptors that are firing to the brain. So it's kind of like a chromatogram, basically. The brain is interpreting the chemical structure of all these different compounds that are coming in, and the brain remembers that pattern. So the next time you consume a product, and it's the exact same pattern of flavor volatiles bonding to the receptors, your brain remembers that pattern, and if you've labeled it as something, mango or peach or whatever, then the brain remembers that pattern. And so this is just kind of visually what's happening. We're smelling food as the odor molecules are coming off the food. They're going up into the brain, up into the high part of the nose, where we have the olfactory receptors. And these flavor molecules are binding to the receptors. And these receptors are G-coupled protein receptors. It's a very common type of receptor in the body. And so basically you would have a certain part of the molecule, a certain chemical structure within that molecule that would bind to a specific receptor. And then that establishes the nerve impulse. And so this receptor is firing a nerve impulse to the brain. And that's one of the signals the brain's interpreting. And it may be interpreting thousands of receptors going off at the same time. And this is just another picture of this. This is from a wine, actually. But you've got all of these different receptors that are firing nerve impulses to the brain. And the brain's got to interpret that pattern. Is it a Merlot? Is it a Pinot Noir? What particular pattern is that? And if you remember the first time you ever smelled a food for the first time, I remember vividly tasting a mango for the first time. I was in the Dominican Republic a long time ago, and I never had a mango. And I remembered that, and I had no label for it. When I first experienced that, I thought it was great because it was sweet, and certainly I enjoyed it. But I didn't know how to label it until somebody said it was a mango. And then my brain remembered that pattern the next time I had a mango. So it's a pretty amazing process. So once you get the odor and you smell the odor, and that's a big part of some foods, not so important in others, but it certainly can be very, very important. Then taste kicks in. And there's 4 basic tastes, as I mentioned: sweet, sour, salty, and bitter. Some people consider umami a basic taste. At least for this talk, I will not do that. And so we detect taste in our taste buds. I think everybody's heard of taste buds. And the taste buds are located on papillae in the tongue. The tongue has a lot of rough surfaces, a lot of structures and bumps all over them. And these taste buds have receptor cells, just like we have receptors for the olfaction. And the sweet and bitter receptors are very similar to the odor receptors. They're large proteins that are embedded in the membrane. And there's 2 or 3 receptors for sweet, and there's about 20 receptors or 25 receptors for bitter molecules. And so a sweet molecule or a bitter molecule would bind to the specific receptor and establish a nerve impulse to the brain. And sour and salty are a little bit different. They go through ion channels. And so basically you have sodium or hydrogen ions that are flowing through an ion or a channel in the membrane that establishes the nerve impulse. So sweet and bitter are detected quite a bit different than salty and sour. So this is a picture, not a very nice picture, but it's a picture of the tongue. And it shows the papillae. So these are the 3 different types of papillae that contain taste buds. So you got fungiform are in the tip of the tongue. The circumvallati are these really big ones that are in the back of the tongue. And the foliate are along the sides. And these papillae are actually large enough that you can see them under very low magnification. You can stick your tongue out with a magnifying glass and actually see these papillae. So they're fairly large. And these papillae contain the taste buds. And so the taste buds are much smaller. And some of the papillae may contain just a few taste buds or may contain many taste buds, depending on the structure. And so these taste buds are either at the surface or down in the valleys of the papillae. So the papillae are sticking up really off the surface of the tongue. And the reason we call these little structures taste buds is because they look like a bud. They actually look like a little onion. It's a collection of cells that shape very unusual. And they have receptor cells indicated here. And these receptor cells end in microvilli. And so the receptor proteins would be located in the tip of this membrane, basically. So this is where the receptors would be located. And whatever's on the tongue and the saliva would be coming in contact with the receptors at this point. And the receptors for sweet and bitter, as I mentioned, are also G-coupled protein receptors. They're very similar to the olfactory receptors. And so a sweet, a sucrose or glucose or fructose would bind to one of the sweet receptors and establish a nerve impulse. And if it's a bitter, we have about 20 different bitter receptors, and the bitter molecule would bind to one of those receptors and establish a nerve impulse. By the way, the reason we have so many bitter receptors is bitter historically in our evolution has been associated with bad things, poisons and things like that. So we have a pretty broad spectrum of bitters that we can detect, basically for protection. And the discovery of these receptors resulted in the Nobel Prize in 1994. And this would be how we detect sodium and hydrogen ions, or salty and sour. In the case of acids emitting the hydrogen ions, it would basically flow through a certain type of ion channel and establish a nerve impulse. And the same with sodium. Sodium would be flowing through a channel in the membrane and establishing a nerve impulse in that regard. So, are there genetic variations in taste? What do you think? I don't know if you can respond, but well, yes, there are actually quite a bit of variation in our ability to taste. Our ability to detect odors also, but especially in taste. And there are some people who have a lot more fungiform papillae than others. And if you have more papillae, you have more taste buds. And so there's certain part of the population that contains 2 or 3 times the number of taste buds than an average person would be. And I don't know if any of you have ever heard the term super tasters, but these are people who contain who have a lot more taste buds than others. And if you have more taste buds, you're going to detect all the tastes much stronger. Sweet, sour, salty, and bitter are all going to be detected much stronger. And you can actually also detect capsaicin and the burn of hot, spicy foods and other sensations like the feel of creaminess of fats. Those sensations are stronger because you have more of those nerves on the papillae also. So the papillae carry other nerves other than just the taste buds. And so this is so strong in some people that it can influence eating habits. And they tend to avoid things that are very bitter and tend to avoid high fats. And so it can really actually influence people's eating habits because they tend to avoid certain foods. And there's several ways to determine if you're a super taster. About 20% of the population is believed are super tasters. And you can actually count the number of taste buds on a small portion of your tongue. And that's how it's actually done clinically, is you count the number of taste buds in a very small area. And that would be how you would be classified as a super taster. And some people have very few, and they would be considered a non-taster. And most people are somewhere in the middle. I happen to be not a super taster, just a medium taster. So now we've got, I think the most important part of our food eating experience is retronasal olfaction, and that's the flavor molecules that are now in the mouth. So the same odor molecules that you smell in your nose coming in from the outside are also being emitted in the mouth. As you start chewing, the flavor molecules break apart and come up into the same system. And so the odor molecules this time come up the back of the throat into the nose, and I'll show you a picture in a minute, and go to the same type of receptor cells, to the same receptor cells, except the brain processes that in a different part. And so the brain knows whether the odor signal is coming from the outside or whether it's coming from the inside of your mouth. And retronasal olfaction is what we normally call flavor. And these odors are learned, as I mentioned a while ago. The brain remembers the templates. How many different odors can you recognize, or how many odors do you have the ability to remember? It could be in the thousands. I don't know how many foods that I can identify based on the odor. But once the brain recognizes this pattern of all these receptors that are being fired, you label it as something and the brain remembers that. And so it's really an amazing process. And then we learn to like or dislike it. And that's a whole nother lecture. But one way to dislike a flavor immediately is if you get sick. So I think everybody's had an experience where you've eaten too much of something or drank too much of something that had a certain flavor associated with it. And if you ever throw up after consuming that flavor, you usually develop a very strong disliking of that flavor. And learning to like a flavor can be associated with if it has high sugar, if it has high protein, if it does something good for you, then we learn to like the flavor. So how we like and dislike things is pretty complex, actually. So this is what happens in retronasal olfaction. We've got the food in our mouth. We start chewing it up. The odor molecules, the volatile molecules are going to come off that food and go up to the same tissue. The receptors are here, the same places from the outside, except the brain knows that it's coming from the inside and is processed in a different part of the brain. So that's a brief overview of how we detect odors and taste, and that's where it all starts. So every time you consume a food product or someone sits down and does a sensory test, that's where it all starts. And now we've got to measure whatever the response we're asking the panelists to give us. So that's where the sensory testing comes in. And there's 3 different types of sensory testing. Discrimination testing is common. Consumer acceptability preference testing is probably the most common type of sensory test that's done in the food industry and in the research that I do. And descriptive analysis is the 3rd general type of sensory test that I'll talk about briefly and give you some examples of all of these. A discrimination test is very simple and very straightforward. The only thing you're trying to determine, are these 2 products the same or are they different? And that's all you want to know. And this is a test that has some very specific applications, but you don't want to do a discrimination test if there's obvious differences between the samples. You're just wasting your time, and you should do some other test that's going to give you more valuable information. For instance, you would not want to do a discrimination test between Coke and Pepsi. Most people can tell the difference between Coke and Pepsi. It would make more sense to describe how they're different or describe which one do you like the best. So usually this type of testing is done where you're dealing with 2 food products that are pretty close, and you really have to determine, are they the same or can people tell a difference? So this is a very easy test to do. It can be done at a very local level. You really don't need a sophisticated anything to do this test. And it's very powerful. If that's the answer you want to know, is there a difference or not, it's the most powerful test you can do to give you that answer. But that's all it tells you. So if you have a significant difference in the end when you do your test, it doesn't tell you which product is liked the best, how big the difference is. It doesn't give you any information other than, can the panelists tell a difference or not? And so some examples of usage of that, one common usage is in ingredient substitution. Food companies substitute ingredients all the time. And I'll use Coke as an example. I don't know how much they substitute ingredients, but let's say that Coca-Cola, Coke has to substitute an ingredient. So they've got one ingredient in their formulation, and due to cost or they've discovered a cheaper source of it for whatever reason, they want to substitute this new ingredient from a new supplier or a cheaper ingredient. But the last thing they want to do is change the flavor of Coke. They learned their lesson on New Coke with that, so you don't mess around with Coke. So you want people who drink Cokes every day to be able to tell you, can you tell a difference in this new ingredient or not? And that would be a very powerful test and one that they may want to know. The same way, a lot of companies will have several suppliers making the same product. And we did a test with this with a fast food restaurant chain in Florida, and they had one supplier of French fries, and that made them very nervous if something happened to that supplier. So they wanted another supplier to make the exact same type of French fry, and we actually did a triangle test or a difference test to show that both of these suppliers could make the exact same French fry. And so product matching is also very important. If you're trying to match a competitor, if you've got a knockoff product and you're trying to match it to a name brand, how close can you really get on that? So product matching is one also. So this has some very specific applications, but it can give you some very powerful information if you really have to know, can a consumer distinguish between these products or not? And the most common test, there are several different types you can use. The most common that we use and is used across the food world is called the triangle test. And you're trying to determine the difference between two samples. Is there a difference or not? And the reason that this is more commonly used is that there's always a chance of guessing the right answer. And so you don't know whether panelists guessed the right answer or whether they actually were experiencing that. So you have to assume that the maximum number of people who guessed did guess correctly. And so in this one, there's only a 33 chance of guessing the correct answer. That's a lot better. Most of the tests, there's a 50% chance of guessing the right answer. So if there's less chance of guessing the answer, that means it's a more efficient test and you need fewer panelists. And fewer panelists is always good, especially in an industry setting. So in the case of a triangle test, you can get pretty good results with 50 or 60 people. And some of the other tests may require up to 100 panelists. And in this test, this is something that you can do very easily. You present 3 samples to the panelists. 2 of those are one type and one is different. So 2 Pepsis, 1 Coke, 2 Cokes, 1 Pepsi. You mix them up in all orders of presentation and give them to the panelists with coded numbers. And you tell the panelists, 2 of these are the same and 1 is different. I want you to pick out the odd sample. It's actually a pretty tough test. And so it actually frustrates consumers sometimes doing this test because they want to get the right answer. And we will often have the panelists come up to us after the test and say, did I get it right? Did I get it wrong? And they want to know. So it's kind of a guessing game for the panelists. And so the statistical analysis of this test is very easy. You count the number of correct responses out of the total. And there are tables that have done the calculations for you. And you go to this table. And based on the number of panelists you have, it will tell you how many correct responses do you need for there to be a significant difference. So it's a very simple statistical analysis based on probabilities, and it works very well. So this is a very common test that we use when we do a difference test, and it's probably the most widely used in the food industry. The other type of test, the other type of test that we do all the time, and is probably the most important type of test in the food industry, and it's probably the most important type of test that I do in our research program. And that's to assess how much people like something, the acceptability, or which sample do people prefer. And so that's usually the bottom line. That's usually the information you want. If you do a triangle test and you establish that there is a significant difference, the next test you may want to do would be a consumer acceptance test. And so now you know they're different. Which one do people like the best, this one or that one? So as you could all imagine, I'm sure you could all think of a lot of different examples of where this would be very meaningful in research and in the food world. And one of the biggest challenges of this that we have in my environment is targeting the consumers. This is a sampling issue. You know, if you have a General orange juice, there may be millions and millions of people in the United States that consume orange juice, and you would like to get opinions of millions and millions of people, but that's just not possible. So you have to select a subset of that. So we're going to select 100 panelists that represents 20 million people to taste that product. You better get a good sample. And so targeting the consumer, making sure the people you ask to do this test are consumers of that product is very, very important. And so you want to use consumers of that product. Sometimes that's a challenge for us because we have a limited demographic on campus. We recruit a lot from students, but also faculty and staff. So we can get a pretty broad group of consumers. But one time we were asked to do a test on Jiffy Muffin Mix, actually. And they wanted people who consumed Jiffy Muffin Mix like once a week and were over 45 years of age. And it took us about 6 months before we finally identified that population. So sometimes the consumers need to be heavy users of that product, and sometimes you just want a casual user of that product. So this is a very important part of this whole thing. You better collect a good sample if you're going to make a conclusion based on 100 opinions. And so there's a lot of examples. I'll give you some that we do all the time. We have a food company here in Florida. It's a beverage company that benchmarks their product against their competitors' products routinely. So they'll go to the market and get some of their product, get some of their competitors' product, and do a consumer acceptance test to determine how is their product compared to their competitors' product. So they do this several times a year. And so benchmarking against a competitor is one very common use of this test. And in research, if you're doing product development and you've got several versions of a new product and you've been doing benchtop testing for a long time, at some point you've got to get it out into the real world and unbiased people and determine which version of this product that people really like the best. And we do a lot of sensory testing with the food company here in Florida, Bacardi actually. And they make all different types of products. And so they may give us several versions of a particular product, and they want to know which one of those are we going to move forward with in our further research. I do a lot of consumer acceptance testing in the research that I do. I work a lot with the plant breeders here at the University of Florida, for instance, and they're developing new varieties of blueberries. We want to know, is the new variety of blueberries they're developing, does it have better consumer acceptance or worse consumer acceptance than existing varieties? So I use this a lot in the research that we do here on campus also. And so either you're asking acceptability or you're asking preference. And so in the case of acceptability, how much do consumers like things? And there's various hedonic rating scales, which I'll talk about in a minute, that we use. And so it's pretty, consumers are very willing to tell you how much they like something. So this is something that people really like to do. And they're very good at telling you which one they prefer. So if you give people two choices, which one of these do you like the best? People usually have a very strong opinion. I like this one the best. And so sometimes we do a preference test where we give people two products. And really what we want to know is which one do you like the best? And so that would be a paired preference. And sometimes we measure acceptability, and then in the end, we force them to pick their favorite product out. And so if you're comparing more than two products, you're doing a ranking test instead of preference. So which one is in first place, which one's second place, third place, and on like that. So usually we're measuring some level of acceptability or preference, which one do people like the best. And as I mentioned, 80 plus panelists or better. It would be good to do a sensory test on 400 or 500 people, but that's just not practical and it's very expensive. And so we usually aim for 100 panelists. And that the number 100 is nothing magical. Usually if you get between 80 and 100, you get pretty good results. I think if you drop off below 50 panelists, you really start getting in la-la land, and you may not be able to make a firm conclusion on that. So more is obviously better. It's just when do you reach a point of diminishing returns, and what's the fewest number of panelists you can get away with is the bottom line. So 100 works for us. We had a real challenge of this. I was telling Amon during COVID. We had to shut down sensory testing for about a year due to COVID. And when we started back, we did not have many panelists on campus. It was all virtual online, and we had so many people working from home. We usually could get 100 people quite easily to do a test. And we were struggling to get 50 to 60. And so that really compromised some of the tests that we were able to do because we just couldn't quite get enough panelists. Okay, so the main thing you're going to determine is how much do they like something on some scale. So the typical scale that's used in the food industry is the 9-point hedonic scale. A lot of you may have done a test involving this. 1 is dislike extremely, 5 is neither like nor dislike, and 9 is like extremely. Actually, there's a verbal descriptor for all 9 numbers on that scale. This was a scale that was developed by the military back in the late 40s, I believe, to measure how much soldiers like rations. And it's been used ever since then. And I would say it's used hundreds of times every day in the food industry. And so this is a very common scale. I'll show you on alternative at the end and some research that we're doing, because this scale is good for detecting differences between your samples, but it's maybe not so good at measuring absolutely how much you like something. And so there's other scales that will give you different information. But that's the one that's primarily used, and the one that we primarily use in our research. So we usually are evaluating 3 or 4 samples. There's a practical limit on this. It's impossible to give people 20 samples and ask them to taste 20 samples and rate how much they like them. It takes too long. Panelists are going to lose interest, and they're going to start getting adapted to the taste and perhaps the odor. So there's a practical limit on the number of samples you can do in a consumer test. And 6 is about the maximum we've ever done. When we do blueberries and strawberries, they're pretty easy to taste, and we can do up to 6 blueberry samples, for instance, in a panel. But that's really pushing the envelope. And if you have something that's a very strong flavor or lingering flavor, or something very hot and spicy, you may have to limit it to 2 samples, and that would be it. It's important that all the panelists rate all the samples. So you need a good experimental design. And so you want to be able to do a randomized complete block where panelists are blocks, and they're evaluating all the samples within that block. And so that's the statistical analysis that you really want to do. And you use analysis of variance on this for rating data and mean separation to analyze the data. And it's very handy. A lot of food companies, and in our research, we use computer data entry. And we have a software set up for sensory testing, and it automatically does analysis of variance, mean separation. and even creates a PowerPoint presentation. So it's very easy to do the data analysis on the modern software that we use. Okay, the last type of sensory test that I would like to mention is one called descriptive analysis. This is used to describe what are the attributes of a sample that differ. You know, panelists or consumers, that is, are very good at telling you why they like one product over the another. If I ask you to tell me which do you like, Coke or Pepsi, or Dr. Pepper, Mountain Dew, whatever, you would have a pretty strong opinion. I like Coke over Pepsi, or Pepsi over Coke. But if I ask you to tell me why and describe why you like it, you might really struggle. Even you as food scientists might. So what is the difference? Is it sweeter? Does it have more cranberry flavor? Does it have more citrus flavor? So now that becomes important, because if you're in product development and you know that one product is preferred over another one, you need to know why, because you have to know what to fix. And so usually you want to know what attribute is the problem. Why is my competitor's product better than mine? The consumers will tell you that, but they may not be able to tell you, is it sweetness? Is it sourness? Is it a flavor system that's not good? What is it? Or maybe it's appearance or texture. So that's where descriptive analysis really becomes valuable. If you have to describe how is my product different from my competitors, or vice versa. And this can include evaluating the appearance, the aroma, the flavor, the taste, and the texture becomes very important too. The texture is very important in a lot of the fresh fruits and vegetables that we taste. Blueberries, tomatoes, it's extremely important. So descriptive analysis usually also contains texture if it's a solid. And so this is a very valuable test. It can give you some very valuable information, but it's by far the most difficult of all sensory tests, because it requires a lot of time. And time is money, and in the food world, these types of tests are very expensive. And so we have a little more time in academia, and so sometimes we will train a panel to do descriptive analysis, but we don't have one trained all the time to automatically do descriptive analysis. And so you'll see in a minute why it takes so long to train a panel. But some examples of when we've trained a panel recently, we had a graduate student a couple of years ago who did a project on hops. And hops are used in beer. I think everybody knows that. But they're trying to grow hops in Florida. And there's big question as to why we're trying to do that, but we are. And basically, hops are usually grown in Oregon and Washington and Germany and Northern Europe. But there's a big demand for hops in Florida, and there's a big demand for hops, period. And so if we could grow them in Florida, it would be certainly, there would be a big market for this. So you really can't ask people to tell you whether a hop is good or bad. And so really you can't do an acceptability test on this. What you have to do is describe. And so we described it based on aroma. That's how the brewers evaluate hops, is they just, smash it up in their hand and smell it. And so we trained the panel to identify the various characteristics of hops. A citrus aroma, there's a pine needle aroma, there's various things that we had, about 10 descriptors that were used to describe hops. And what we found was that the hops growing in Florida really did not have much of any character compared to the hops that were grown in Washington State and Oregon. Another time we did descriptive analysis, we did it on a particular wine that's produced in Florida. And so there's a variety of grapes that was developed at the University of Florida called Blanc du Bois. And it's a long story, but Blanc du Bois is grown in Florida and Louisiana and Texas. And those are really the only three places. So we felt like the Blanc du Bois grapes that were being grown in Texas and Louisiana and Florida could be quite different and make quite different wines. And in fact, they were. And so instead of asking people, which wine do you like the best, or can you tell the difference, we wanted people to describe the wines produced from this grape in Texas versus the same grape grown in Florida and Louisiana producing wines. And so we trained a group of panelists to evaluate Blanc du Bois character, both aroma and flavor in the mouth. And we came up with a list of about 12 to 15 different descriptors that describe Blanc du Bois. And what we found in the end is that the wines produced in Florida were very green and grassy, and the wines produced in Louisiana were very fruity, and the wines in Texas were kind of in between. So it gave us some valuable information to move forward with. So in a descriptive analysis, you've got to use well-trained panelists, and I mean well-trained. And so there's several different approaches to doing this, but you start with a small number of panelists. You can't train 100 people. So you start with 10 to 15 people that you're going to train. And usually you start with providing a wide range of the product. We also trained a panel on orange juice many years ago, and we gave panelists every orange juice we could find in the market, and they selected important attributes. And so basically they selected like 12 to 15 different attributes that were present in these different orange juices. And this took maybe 5 to 10 different training sessions. So this took 5 to 10 hours just to be able to identify the attributes that are in these different products. Then they go through a series of trainings and subsequent meetings where they're trained to accurately identify this attribute. So if you say something is a fresh orange, we would basically squeeze fresh orange juice, and that would be the standard we would use for fresh orange attribute. And if it were cooked attribute, we would use an orange juice that had been heated extensively, and that would be the cooked flavor. So you've got to train people in maybe 10 sessions to accurately identify those attributes and consistently rate the intensity of them. So that's where introducing references becomes very important. For the basic taste, there are guidelines. You could give somebody a 5% sucrose solution, and everybody agrees that we're going to give that a rating of 8 on a 1 to 15 scale. So you've got to find a way to consistently rate the intensity of each attribute and then identify it. So once you have your panelists trained properly, and it may take 20 training sessions to get to this point, then they're going to actually evaluate the samples, and that's the end product, what you're trying to do, and they usually do it in replicate or triplicate even. So this takes a lot of time. Even in academia, it takes us a long time, and in industry, it takes a long time. And a lot of people, if you're in the food industry and you want your product to undergo descriptive analysis, a lot of food companies will use a private lab. There's several that do descriptive analysis, and it's not cheap, but it's sometimes the easiest way to get this done. Okay, are there any questions? I don't know how to take questions. Are there any questions at this point? I'm hearing nothing, so we'll move on, and then I will stop sharing my screen and we'll take questions then. So we've got about 15 minutes or so. So I want to talk a little bit about some of the research we're doing, and I can speed up or slow this down any way that we really need to go. But one of the things that we're really interested in is, can taste influence odor perception, and can odor perception influence taste perception? And so it goes both ways. And so we've done a lot of work on how can taste modify odor perception. For instance, can sweetness influence odor perception? So if you increase sweetness, does that influence the flavor intensity you're getting from that? And if you can enhance sweet without adding sugar, what does that do to the odor perception? And so we've done several research projects on this, and we use Miracle Fruit and Gymnema. And I don't know if any of you have heard of this, probably so. Miracle Fruit is a fun thing. It's kind of a fad. Both Miracle Fruit and Gymnema are classified as dietary supplements, so they're not foods. But Miracle Fruit is a small red berry that's produced in the tropics, and it's usually freeze-dried and turned into a tablet. So you can buy Miracle Fruit tablets on the internet. You can buy them from Amazon. They're very commonly available. And what Miracle Fruit does, once it dissolves in your mouth, it really enhances the sweetness of any food containing an acid. The first time I did this, I was shocked. I really expected the influence to be very small, but it's a dramatic influence. And so, but the food has to contain acid. So a strawberry, a lemon, something like that. And so if you consume the Miracle Fruit and then consume a food that contains acid, it tastes extremely sweet. So we also measured, well, how much does the flavor go up? And I'll show you some data of that in a minute. And certainly the flavor intensity did go up. As sweet goes up, the flavor intensity went up, and you're not increasing the sugar. You're just telling the brain that there's more sweetness there. The same with Gymnema, only Gymnema goes the opposite way. Gymnema is not much fun. Miracle Fruit is a lot of fun to do a party with and play with. Gymnema is not so fun. And Gymnema is an herb that's dried, and usually people make a tea out of it. So you can make a tea out of this, brewing it in hot water, and you drink this tea, and Gymnema totally blocks sweetness for about 10 to 15 minutes. And that's not a very pleasant thing, but we can now take sweetness in the opposite direction. So Miracle Fruit will enhance sweetness without affecting the sugar, and Gymnema will lower sweetness without reducing the sugar in any way. So you're just telling the brain that the sweetness went up or down, and what does that do to odor perception? So if you go in the opposite direction, does flavor perception also go down? So I'll show you some data of that in a minute. And does it work the other way? Are there certain odors present in food that can influence taste perception? And we've discovered this. A lot of people have discovered it over the years. But when we did a large project on tomatoes, where we tasted maybe 100 tomato varieties over the course of 3 or 4 years, we were seeing tomatoes that tasted very sweet that didn't have a lot of sugar in them. And so we started doing statistical analysis, and we had measured sensory response, and we had measured all the flavor volatiles and the sugars. And so through multiple regression analysis, we were able to identify certain volatiles in these tomatoes that enhanced the perception of sweetness over and beyond sugar. And so we got very interested in that. And so that was all based on statistics. And then we started doing the same thing with peaches and blueberries and strawberries and some of the other fruits that we were working with at the time. And we were able to continually identify flavor volatiles that we thought increased the perception of sweetness. The same with saltiness. We think we actually have some ongoing projects with that now. Are there certain volatiles that will influence the perception of saltiness, particularly increase the perception of saltiness? And so I'll talk a little bit about that in a minute. But basically, all of this was statistics. And so we were statistically proving that, but the real thing is can you apply this? And so you can buy most of these flavor volatiles food grade. And so we went out and bought as many of these flavor volatiles as we could find and added them to a sucrose solution and determined whether sweetness went up when you added the flavor volatile or not. And the same with salty. If you added certain volatiles to a salt solution, would the perception of saltiness go up? So this is ongoing research, and I'll talk a little bit about this in a minute and show you some data. And the last thing I'll tell you about a little bit, if we have time, will be a new scaling method for measuring liking. Again, the 9-point hedonic scale that I mentioned earlier is a great scale, but it has certain limitations, and I'll point out those limitations in a minute. Okay, so here's some data from the study we did on miracle fruit and gymnema. So basically, this is for strawberries. Strawberries have acid in them, and so miracle fruit should basically make strawberries taste a lot sweeter. And so we smelled them first. There's no difference in odor. And then this would be the control. And then so the strawberry before anything. And then after they consume miracle fruit, this would be the sweetness. After they consume gymnema, this would be the sweetness. So you can see that the sweetness goes up quite a bit after they consume miracle fruit. And when you consume gymnema, the sweetness goes down quite a bit. So this is what we knew. This has already been shown many times before. But what we didn't know is what would happen to the flavor also, the retronasal flavor. And so basically the same thing happened. As you increase sweetness by miracle fruit, you also increase the perception of flavor. And when you decrease sweetness through gymnema, you also decrease the flavor intensity. So it goes both ways. If you increase sweetness, you increase flavor. And if you decrease sweetness, you decrease flavor. So we felt like flavor was being influenced by sweetness. And then sourness went down in strawberries once they consume miracle fruit because as sweetness goes up, sweetness tends to suppress sourness, and so you would expect sourness to go down as well. So this shows something interesting with maple syrup. Maple syrup is very neutral. It doesn't have acid in it. And so miracle fruit should not have any influence on this. And in fact, that's what we showed. So basically, if you look at sweetness, after you consume miracle fruit, nothing happened to sweetness. But when you consume gymnema, the sweetness went away dramatically. And this was really a dramatic drop in sweetness. And it was like tasting slime in the mouth. You could really get the slimy feeling, and it was just nothing there. It was really an odd sensation. And so as the sweet went away, so did the maple flavor. The maple flavor was reduced considerably. And so this is one where we could not increase sweetness, but we could take sweetness away, and it really dropped the flavor intensity. So those are just two examples that we did. There were other examples also. If you ever do this for fun with gymnema, I would encourage you to do lemons. It makes lemons taste like lemon candy. It's really amazing. And if you like grapefruit, but grapefruit is too sour, grapefruit tastes wonderful after miracle fruit. So this is a fun thing to play with in a demonstration also. But we used it in some of the research we did. And this shows can volatiles enhance sweetness. We're getting close to the end here. So in this experiment, this is where we took a 2% sucrose solution, which is not real sweet, but it's certainly certainly you can perceive sweetness. And we added 11 volatiles that we had identified from tomatoes that enhance sweetness. So these were identified by statistics. We bought them and we added them to a 2% sucrose solution. And we did the same thing with 24 strawberry volatiles that we had identified. We bought those. We added those volatiles to a 2% sucrose. And then we combined both the strawberry and the tomato. So we had all 35 volatiles and we added that to 2% sucrose. So as you can see, basically, and what we did what's called the nose bump technique. And this is something fun to demonstrate also. If you plug your nose up and taste the food, you can detect the taste, but you totally block retronasal olfaction. If you've got your nose plugged, you don't have any air currents, and the flavor volatiles can't get to the olfactory receptor. So in fact, you basically block flavor. And so if you consume a product with your nose plugged and rate sweetness, and then as you're swallowing, you open your nose, you get the rush of volatiles up to your olfactory tissue, and you can detect the flavor. And so once that rush hits the nose, does the perception of sweetness go up? So that's what we ask our panelists to do. Hold their nose, basically rate the sweetness, then open their nose and rate the sweetness again. And that's what they did. And so you can see when you had a 2% sucrose, nothing happened after you open your nose up. But in the case of the volatiles, we saw a significant enhancement of sweetness once the volatiles hit the nose. The same thing with the 24 strawberry volatiles. And when we mixed them all together, we got even a stronger response. And so now we've identified up to 64 volatiles that we use in a mixture to enhance sweetness. And it works very well, actually. And we're doing the same thing with salty now, only basically we've got a dilute salt solution, and we're adding various flavor systems and volatiles to the sodium solution to determine whether saltiness goes up or not. But this nose bump technique that we use is quite valuable, actually, and it's quite fun. It's an easy demonstration you can do on yourself. Just eat a piece of candy or take a sip of orange juice with your nose plugged, and then swallow it and open up, and you can then distinguish between what taste is and what flavor is, or retronasal olfaction. And the last thing I want to mention is just this, and then I'll stop and open it up for questions. This is what we call the Global Hedonic Intensity Scale. And I have a good colleague in our department, Linda Bartashuk, who basically brought this scale to us. And it's one that she's developed over the years and tweaked over the years. And instead of just measuring something on a 1 to 9 scale, the problem with that is that if you wanted to compare across people, are females really liking this product more than males? Or is a super taster liking this product more than a non-taster? You know, if everybody gives it a 6, you may have 5 people that give it a 6 or a 7, but they're all experiencing different things. You're just using parts of the scale differently. And so that's a common thing that consumers do. And so if you want to compare across people and get more of an absolute rating of how much do people really like this food product, this is a scale that we think will work better. And we've done some research on this. So this goes from 0 is neutral. 100 is the most intense liking ever experienced. And so we ask the people, what is the most intense liking you've ever experienced? And it's usually falling in love. Sometimes it's a religious experience. The Bears winning the Super Bowl, the Gators winning the national championship, whatever the top of your world is, that's what 100 gets. It's hardly ever food. Nobody has food as the most intense liking. So that's very rare. And that's very important that food is not at the top of the scale. And then the most intense disliking ever experienced is usually death or something like that. So it's something very negative. And so we ask people to define these two extremes of their hedonic world. So this is their entire hedonic world. And then we ask people to rate, tell me how much you like your favorite food and how much you dislike your least favorite food. And we've done this on thousands of people. And the average rating for liking a food is about a 60. So it's there's people like a lot more things better than food, basically. But it's usually about a 60. The most negative food is around the negative 30 because we just don't consume many foods that are really bad. And so now people define their food world. The food world is here to here. And then if we're doing strawberries that day or orange juice, we will ask people, okay, what's your favorite orange juice of all time and what's your least favorite orange juice? So now they define this is my orange juice world is here to here. And then we actually give them the products and ask them to rate the products that they're evaluating that day. And what we found on this scale, and we published a couple of articles on this, is that people can still distinguish between samples just like they could using a 9-point scale. But now we feel like we can identify, do super tasters really like this product, this orange juice, more than non-tasters or vice versa? So we feel like we can compare across people a lot better and pick out groups of consumers that may have things in common. So with that, questions, comments, let me stop sharing my screen.
Okay, thank you, Dr. Sims, for the wonderful presentation. Audience, floor is open for the questions.
I hope I didn't go too fast or cover too much, but
No, it was great.
Questions, please, or comments.
Yeah, Uday, you can go ahead.
Hi, Dr. Sim. It's a really nice presentation. I really like it. I'm just wondering if there's any difference between train a non-taster and a super taster as a panelist. And also, is that training for the panelist like very similar to train a person to work with the GC Ultra authortometry?
I didn't hear that last one. I'll address the first one though. Is so certainly when we're recruiting panelists, in some of the panels that we do, if it's just an average food panel with a food company or most average research that we do, we just recruit people and we do not screen for whether they're a taster or not. So we just assume that there's going to be a certain percentage of that population that are non-tasters, certain super tasters, and most of them are medium tasters. And that's pretty much what the real world is going to give us anyway. There are some situations, though, where we would want to know that. And so in that case, there are ways that you can screen people, and we usually give them a very strong sucrose solution or a very strong bitter solution, usually quinine, and ask them to rate it. And so the people who rated it like 90 on the scale or higher, we usually have a criteria we use for classifying someone as a super taster or not. I have a good story. It takes too long to tell it, but when we first were doing this, there's a way to measure super tasters using a chemical called PROPE. And we were going to use that, but we were not allowed. It's actually a drug, and we did not have IRB approval to do it. And for us to use it, we were actually going to have to prove whether females were pregnant or not for them to participate. And obviously, that was something that we were not prepared to do. So, the bottom line is sometimes we try to know whether they're super tasters or not, and then we can pick out that data and look at their data separately from a non-taster. What was your second question? I didn't quite catch the second question.
Would train a panelist very similar to train a person to work the GC olfactometry?
Okay, so train panelist versus GC olfactory.
Mm-hmm.
So I'm not that familiar with GC olfactory. I know what it is. I don't use that routinely, but normally you have to be trained a little bit on that. It would not be like training a panel for descriptive analysis, but sort of like that, yes, because I've done it before. It actually, you've got to be able to describe the odor coming out. So you need a list of descriptors. Does it smell like shoe leather? Does it smell like a peach? So you need to be able to accurately describe that. And some people are just naturally better at doing that than others. And so you've got to be able to describe what you're smelling. And often they'll have a list of terms on the side that they can automatically look at that term and decide. And then they have to rate the intensity of it in some way usually. So yeah, that would require some training, but not quite the same training maybe as a descriptive panel. That's a good question.
Thank you. Thank you so much.
So I'll ask a question. Thank you, Dr. Sims. That was a fantastic presentation.
Thank you.
And I'm sure you've gotten this a million times. So I actually have two. One has to do with, of course, COVID's, the loss of smell and taste and, the relationship to having a viral infection.
Right.
And then second to that, losing one's smell, or rather having phantom smells, and even I'm trying to think of the other term, but it's not phantom, but it's where you'll smell something, but it's not the correct smell, and it might even smell very tarish or very chemically driven. And they say that that's associated with Parkinson's disease. So is that at the level of the olfactory cells, or can you explain a little bit about that?
Those are good questions, and some of that's out of my area of expertise. I have a colleague, Linda Bartashuk, who works that, and we have a Center for Smell and Taste, and they do a lot of research in this area. I'll address COVID first. So most of you know that one of the common symptoms of COVID is a loss of smell and taste, temporary for about a week. The worry is that it's more than temporary. So the worry is that you've damaged your nerves by that virus, and your ability to especially taste may be permanently impaired. We were worried about this when we started sensory testing again. And so we worried if somebody came in that had COVID and didn't know it, would their data be any good, basically? And probably it would not have been very good. So number one, we just assumed that if somebody knew they had COVID, they were not coming in to do a sensory test. So we were hoping that was the case. And then if somebody had COVID and had recovered from it, we were just assuming that they would be like a normal person. In fact, they might not have been normal. So we perhaps during that time we might have had some sensory testing data that were influenced a bit by that. I would say though the normal genetic variation between people probably makes a bigger influence than the small number of people who would have had COVID. So I would say that the normal variation in people's ability to taste and smell was probably had a bigger influence than whether they had COVID or not. But I have no way to prove that. The second one was when you have a loss of smell or loss of taste. Either way, it could go either way. If you have loss of smell, though, due to whatever reason, you can have nerve damage. There's a couple of reasons. It really influences people's quality of life because if you can't detect retronasal olfaction, foods just taste awful. And so it really is a horrible thing. And how can you overcome that? And actually, there's people looking at there some things that, for instance, if you could influence, if you lose your sense of taste, could you basically overcome that by adding a certain volatile, or could you add something that would improve people's ability to detect odors? And there was a grad student here who developed a test, an early test for determining Alzheimer's, basically, and it was in kind of their loss of smelling in their nostrils. And so she had developed what she called the peanut butter test. I can't remember exactly how it went, but she had people smelling peanut butter at different distances from their nose. And she correlated that with the onset of Alzheimer's, basically. So that gets out of my area really quick, and I really don't know enough to answer that accurately, to be honest.
Right. No, that was wonderful. Thank you. Appreciate it.
Those are good questions, though.
Okay. Any other questions from the audience? Okay, Rakesh, you can go ahead.
Hello, Professor Sims. It was a great presentation on the sensory of food.
Thank
I'm
you.
a student at IIT, and I just wanted to ask you a few questions. Firstly, you spoke about the olfactory proteins in the start of the presentation. So I just wanted to know, does aging affect the olfactory proteins in a positive manner or in a negative manner? Like as a
So
person
how does
ages?
aging affect our olfactory perception?
Yeah.
Well, again, that gets out of my area pretty quick, but it definitely does. I think that the general consensus is that our sensitivity to odors slowly goes down a little bit, and maybe taste, probably not as much, but maybe even some will taste. So again, there's probably so many generic variations and so many things that could happen to you as you got older that could accelerate that problem. And so I don't know exactly what the exact relationship is between aging and what happens to the olfactory cells. But I think in general, our ability to detect odors, our sensitivity to odors, and perhaps taste decreases some as we get older, unfortunately.
Thank you so much. Also, regarding the taste panelists or the super tasters, do they do some particular things to continue maintaining their odor and the taste receptor profile, like over the time period? Like considering you as being an expert in this field for so many years, like you have a particular lifestyle or a particular set of rules to do's and don'ts to continue doing that.
Well, certainly people who are super tasters tend to avoid foods that are bitter. And Dr. Bartoshuk, my colleague, has done a lot of research in this area with the nutrition community, and they've actually found that if it alters the diet, if you avoid broccoli and a lot of vegetables have bitterness, and so if you avoid that vegetables, how does that relate to the consumption of fiber and consumption of certain vitamins? And so certainly it could impact that, because if it's really bitter, and people avoid alcohol and things that are real burning that are super tasters. And non-tasters tend to consume more alcohol, for instance, because it's not as burning. So alcohol would burn more of a super taster. So it definitely can influence what you like, and you could probably overcome it. For instance, if you were a super taster, you could probably learn to like coffee. Coffee's bitter, but by golly, I want coffee. And so you could probably force yourself to learn to like coffee, even though it was really bitter to you. You might add a lot of sugar to it. So I suspect that people who are super tasters will continue throughout their life to avoid certain things, just naturally, probably. Would be my guess.
Thank you so much, sir. Thank you.
Yes.
Supriya, you can go ahead.
Hello, Dr. Sims.
Hello.
Yeah, we learned about electronic nose and electronic tongue in Aman ma'am's lecture. So I
Yes.
wanted to know how is that compared to a regular sensory panelist? Like how the two compare?
Wow, that's a good question. You know, there certainly would be correlations. You know, so I'm not an expert in the electronic nose or the electronic tongue. So the electronic nose is going to have all these sensors that detect different types of chemicals. You know, water and all these chemicals. So certainly, I think you can train the electronic nose as done to detect certain things. You know, I remember early on we were working some with Coca-Cola, and this is when Amon Dr. Balaban was here, and working on he did a lot of work on the electronic nose. And I asked Coke if they were ever using it, and they used it to detect whether in Europe, they use a lot of returnable Coke bottles, and people put all sorts of crazy stuff in Coke bottles. And so they wanted to determine if gasoline had ever been in a Coke bottle. Apparently, that was a common thing to put gasoline in a Coke bottle and then return the bottle, which sounds crazy. But they could train the electronic nose to detect whether they had gasoline in that Coke bottle or not. So certainly, I think you can. How that would correlate exactly with the brain. The one thing that's missing with the sensors is the brain. You know, so you've got the sensors that may detect the same thing as some of our sensors, but how is it processed? You know, it's a statistical analysis versus the brain. You know, so certainly Amon could tell you a lot more about this than I could. In the same way with the electronic tongue, I even know less about. But I heard a presentation at a meeting several years ago, and they were using something that's kind of like an artificial sweet receptor. So it's kind of like an artificial sweet receptor, and they were using it to screen potential natural sweeteners present in plants. And so they felt like it was a much more efficient way to screen hundreds and hundreds of different compounds than asking panelists to do that. So there are certainly some good applications for both of those. How that exactly correlates with what a human is detecting is something that I really probably can't answer that well, but they both have specific applications. It's hard to duplicate what goes in your mouth, your nose, and the brain, and that's a tough crack right there.
Thank you.
Good question, though. Very good.
Okay, Rakesh, you can go ahead.
Oh, so the last question I had is over the time period, many people tend to like food which they did not like earlier in part of the life, maybe because of the experience or something other than that. So can you like comment on what this kind of perception change makes like sense in the sensorial background?
Say it again now.
Over the time period, many people of different age group tend to like something which they did not used to consume in the past because of some experience or something. So
Right.
this kind of change in the mindset happens due to like something in the sensorial background, or it's just the life experience altogether.
That's a very good question. How we learn to like things, and I think we all have experienced things where you like something now more than you used to, or you like something then you don't like it much anymore. And so there could be a lot of reasons. It could be peer pressure. It could be just repeatedly consuming something. For instance, the first time I ever had a lime flavored Coke, I like lime. I like lime flavor, and Coca-Cola had a lime flavored Diet Coke, and I loved it. So I tasted the first time or two, and I said, "Oh, this is really good." And I kept consuming it, and eventually I just quit liking it. It got to the point I didn't like it anymore, and I don't know whether I was detecting something that I did not detect at first or what. So certainly our ability to like things change over time. I mean, the first time you drink coffee, most people hate beer. First time you have a beer, it's like, beer is awful tasting, and you drink enough, you begin to like it a little bit. So I think that's true for a lot of foods. It's just maybe your life experience becomes broader, and you learn to like things or learn to dislike things.
Thank you, sir.
That's
Thank
where
you.
that learning of how to like things is a whole area of psychology that I'm really not that familiar with.
Okay, Supriya, you can go ahead.
Yeah, I wanted to know that as a like if someone is a sensory analyst for like a tea or a coffee, so like when they burn their tongue like by sipping a very hot tea or something, so what happens in after that? Like, do they regain the entire receptors, or is there any damage that they have to take particular care about?
That's a good question. I mean, the people who do work with coffee, and I'm assuming they do it hot. I don't know how hot it is, but if you were tasting something hot enough to really burn your tongue, I'm sure it would have some type of temporary effect on your ability to taste. I don't know whether there's any long-term effect. If you just repeatedly we're drinking something day in and day out that's hot enough to really burn your tongue and cause pain, I don't know whether there'd be permanent damage or whether it's just a short-term recovery. So I really don't know that, but that's interesting to know. Certainly when you burn your tongue, you tend to avoid putting anything else on it, so but if you repeatedly, I would say though, a bigger danger of repeatedly doing coffee and tea is getting adapted. So if you consume something over and over and over, I do some wine judgings, and we're asked to taste 100 wines in the course of a day. And you get so adapted to things, and your taste and your olfactory system just kind of gets overwhelmed, and you really lose some sensitivity. And I would worry about that more than maybe burning it. But certainly, I don't know what long-term effect burning your tongue would have, but I'm sure it has a temporary effect.
And sir, is it possible to lose a particular like sense of taste? Like maybe some people cannot taste sweet, but like after having a lot of sweet, like is it possible that you can lose a particular taste, like in the tongue?
I think that's true with olfaction. I think you can maybe lose certain things. I believe though, if you have some damage to your tongue or nerve system, that you would lose all taste basically. So I don't know whether you can selectively lose sweet and maintain all the rest of them. So I would suspect that sweet and bitter definitely go together, just on how they're detected. But I've never heard of somebody losing only one taste and maintaining the rest of them.
Okay, thank you.
But anything's possible, I would assume.
Thank you.
Yes. All good questions. You guys have asked really good questions.
Okay. Any further questions before we thanks Dr. Sainz for this wonderful presentation today?
Thank you. I've enjoyed being with you. I enjoy talking about this. So
No, we really enjoyed it.
anytime.
Yep. Alrighty. I hear no more questions. We would like to thank you again, Dr. Sainz, for this wonderful presentation. We learned a lot about sensory. Mostly students, they get exposed to the sensory analysis side. Thank you so much for joining us today.
You're very welcome. I enjoyed it. So,
Yep.
bye, everyone.
Bye-bye. Have a good day. Bye-bye.